Hello and welcome to our attendees and to our seminar on optimizing single-cell RNA sequencing. We are happy to discuss with you today on how to implement a low-cost, low-consumable, streamlined workflow that is enabled through reaction miniaturization. We are going to explore data from a comparative study and showcase technology that enables high-throughput, miniaturized library preparation.
All right, so what I would like to do to start is describe some of the seqWell technologies that serve as the basis for everything that Dave is going to talk about in the second portion of this webinar. So to begin with, what I want to describe is our foundational technology, which we refer to as plexWell. And in this particular case, we are a library prep company, which all of our kits are based on TN5 transposase-based chemistry. Some of you may refer to that as tagmentation and are familiar with it from some other products out in the broad NGS world. What makes our technology different is in what we refer to as a true multiplexing library platform, in which our methodologies allow us to make normalized libraries very quick, very easily, and from a very large number of samples. And when I mean a large number of samples, I mean being multiplexing capabilities up to 2,304 at this moment. But yet this technology allows you to quickly move from 96 samples in a plate at a time down to a single tube in a pool that then you can do the subsequent steps very easily, very quickly, while still incorporating that normalization across about a 10-fold range of input amounts using the chemistry. And at the right of this slide, you see just kind of a graphical depiction of what happens, in which our base plexWell technology is a sequential tagging technology in which we first, in a very controlled method, tag with the I7 barcode in all of that particular end of an Illumina-based library. And then sequentially after that, we would pull down and then add the I5 barcode end of the technology and the libraries in a very simple, straightforward fashion that makes for a very quick workflow, and at the end gives you all the barcoded libraries that you need. And the result of that is something where, from a workflow perspective, because we're pooling from 96 samples down to one tube very quickly, it enables much less consumable usage as well as a faster workflow. And here we're comparing plexWell, the base technology, to NextEra XT, or a ligation workflow, which is typically more than 5 hours to get from those samples all the way through to a library that's ready to sequence. With the plexWell technology, we've reduced that down to about 3.5 hours. And as you're going to see with some other technologies, we're reducing that even farther. With that, however, we took that base technology and started to think a couple of years ago and released this product in 2021, in where we were thinking about single-cell RNA-seq applications, where we wanted to be able to look at full-length transcript type analysis of RNA coming from single cells. And I think some people, when they think of single-cell RNA-seq, they tend to think of the typical 10x type application, in which you are looking at a very unbiased set of data across a big population of cells. You're still looking at the single cells, but you're doing it across a large population. And really what you're doing is simply counting one end of that transcript to understand a broad distribution and relationship between those transcripts, to understand which ones are being expressed at higher level, lower levels, those type of things. What you don't get out of that is the ability to dive deeper. And with single cell full length transcript analysis, in which is what we do with the rapid single cell kit, you can go back and actually investigate different specific cell types of interest through sorting, as well as probably much more importantly, is able to look at really what's happening in your transcript. So you're not just counting the ends of it and saying, well, these are all the same. You're able to actually look at the SNPs, you're able to look at the splicing and the isoforms and everything that's happening with these transcripts in a way. And what you really want to think of full length transcript analysis is as an ideal complement to a 10x like counting methodology. So you look at a very broad picture first, and then you dive down and you start to really try to understand what's happening and understand if there's multiple transcripts, if there's multiple isoforms, anything like that. So it's an additional layer of data that complements what many people do now with their analysis. And looking at this particular kit, just to kind of talk about how we've put it together, it really is a product that is integrated from, the point of having your sorted single cells all the way through lysing those cells, generating the cDNA from them, and then taking that to full-length RNA-seq. So you're actually going from the cells to your sequenceable libraries through this product in this process. And we developed this process in collaboration with Dr. Simone Pacelli from Basel, who actually was one of the, kind of early innovators in all of this type of technology and everything that came along with it. And from a general workflow, you see at the bottom, you've got your single cells that have been dispensed, and then you're going to lyse those, and using a template switching chemistry, you're able to generate that full-length cDNA from your transcripts. From there, those samples and that cDNA is taken through a version of our plexWell kit that we've designed specifically for low input amounts. So we're taking, our base technology plexWell, we've modified it to know that there's only certain amount of input going into it. And so we've designed the chemistry and the tagging strengths and everything else in order to allow that, while still maintaining that normalization and that high degree of multiplexing that we've talked about before, to then generate that single pooled library for you that's ready to go onto your sequencer. And a quick comparison of the workflows as we think of them now. So SmartSeq2 is kind of the protocol and the method that really is kind of the gold standard, or has been the gold standard over time. And we wanted to enable something that gave you just as good or better data, but yet significantly shortened what is a pretty tedious workflow in order to make this work. And you'll see on the left-hand side, SmartSeq2 is a 2-day-ish workflow with a lot of steps, a lot of master mixes or other things that you need to put together to make this happen. And everything works, again, in this 96-well type application and everything else that goes into it. Whereas ours, we've been able to condense down into a single day workflow for for getting these samples ready to go on to and make your libraries from there, with simpler components, simpler things that you have to mix together, shorter incubation periods, everything else that goes along with that, in order to make this a less onerous process for the user of it. But at the same time, a better workflow does you no good if you don't get good results, or at least as good or better results than you already had. So hopefully, without going into great detail with this slide, but what I can tell you is that our single cell rapid kit has been demonstrated to be able to generate higher sensitivity as far as the number of detected genes versus SmartSeq2 or 3, regardless of the sequencing depth that we found with that. And then we're showing, a higher percentage of uniquely mapping reads, which combined with that higher sensitivity allows for you to potentially have to use less sequencing bandwidth on your instrument, and therefore reducing your sequencing cost and any wasted reads and everything else that would come along with having to over sequence in order to be able to get what you need from there. And then we're also showing, very nice uniform gene coverage and a low percentage of things mapping to areas that are not desired, i.e. just ribosomal RNA, anything else like that that you're not interested in. You're interested in the transcripts. And then lastly, as we talk about the single cell rapid, again, talking about our base technology, the normalization aspect that's built into all of seqWell's technology. And this is a situation where by bringing your samples into a very broad, again, about 10-fold range of, input DNA, or in this case, cDNA, the system and the product in general will normalize your read counts across that, again, by using that very controlled tagging step at the beginning. And what you can see in green is a situation where the same samples were put in with plexWell rapid single cell versus a SmartSeq 2, and you can see just the huge variation that comes with SmartSeq 2 for a situation like this in read depths. And you'll be over sequencing some of them, you'll be under sequencing others, and you won't get that consistent coverage across all of your samples. Now, with that in mind, I mentioned before, we're always pushing towards trying to understand, how we can improve the overall workflow. And this goes towards the heart of some of the things our next speaker, Dave, is going to talk about. I want to quickly introduce a new product that we have in early access right now that comes into as part of Dave's work here, and that is the ExpressPlex library prep kit. And this is something that we have designed, whereas I talked about like 3.5 hours for the library prep portion of any of our plexWell technologies, we've been able to condense that down into 90 minutes while still maintaining that high level of multiplexing that we talked about and the normalization features as well. But it is something that we've designed with support of automation and flexible reaction volumes, those type of things going forward. So the workflow for this product in general is, as you see here, both graphically on the left and a little more literally on the right here. But essentially, the product is simply 2 plates. So you've got your indexing or tagging reagent plate on the left here, and then you have a ready reaction mix plate that you see in red. And you simply transfer 4 microliters of your tagging reagents into the ready reaction mix and 4 microliters of your DNA input into that ready reaction mix, seal it, put it on the thermocycler, come back in an hour, and then pool into 1 tube and do 1 single cleanup. And that is your workflow to create 96 libraries very, very quickly. So as I said, this is something that's in early access now, and we were starting to think about how can we do more with this? What's next with that? We designed it for DNA to begin with, but the question quickly became, can we apply this technology into easier workflow for RNA-seq, cDNA inputs, those things? And we wanted to look at both single cell and potentially bulk cell RNA applications. And we absolutely believe, and Dave will demonstrate the possibilities of some of these things right now. And we also said, well, how can we automate and potentially miniaturize this chemistry in order to enable time and cost savings? So that's where I'm going to stop and actually transition this over to our next speaker, Dave Rawling. And Julie, I don't know if you're going to do a quick introduction there or not, but so the idea is this is the base technology. Now Dave's going to show you how it all actually works together.
Great, thank you very much. Hopefully everyone's hearing me okay. And as Curtis just mentioned, sort of following on from what he told you about seqWell's general capabilities, I'm going to be talking through some of the data we've generated working with DISPENDIX technologies to build RNA-seq workflows designed to kind of try and bring this full transcript RNA sequencing method that we use further into the high throughput space. Great. So as Curtis, I think, already alluded to very nicely, we can kind of broadly break down transcriptomic RNA-seq approaches into two families. So the first set of approaches that you see on the top of this figure here generate full-length cDNA molecules, and then they basically index and recover information from the entire body of an mRNA transcript. And of course, we call these sort of full-length or full transcript methods. The second set of approaches are focused on tagging and indexing transcripts at either the 5' or 3' ends, and they recover information primarily or exclusively at transcript termini. There are, of course, advantages to both of these approaches. So obviously, capturing sequence information from the entire body of the mRNA transcript allows you to interrogate many things that you're not going to see about the RNA or that will be missed when you're only looking at the ends, including variants and isoforms and so forth that Curtis already mentioned. Another interesting wrinkle is that full transcript methods actually tend to be a little more sensitive at just identifying a higher number of genes than the end counting methods, which I think adds an additional layer of value. So basically, I think with the full transcript methods, you tend to get more information per cell or per sample. By contrast, one of the major advantages of these end counting methods is efficiency. In particular, I think by continually providing ever more automated solutions, groups like 10x Genomics have enabled higher and higher throughput of cells per experiment with less and less hands-on time, which of course translates to more information being gathered on sort of a per unit cost of time or resources. Even if that data isn't necessarily as rich at the single cell level. Of course, I think very few people will argue that less information about the mRNA transcript is inherently better, and would rather say that sort of the utility and value, a lot of it in these end counting methods comes from sort of the ability to do them at high efficiency. And so I think one of our goals at seqWell is to try and bring some of these higher efficiency methods to bear on full transcript approaches, such that in the fullness of time, researchers will no longer be forced to kind of make these trade-off decisions between sort of more data per cell or more data per transcript and sort of more cells or more transcripts run. So again, as I think Curtis already alluded to, the plexWell single cell rapid kit, which we've used as sort of our baseline for undergoing these studies, is based on a switching mechanism family of full transcript single cell RNA-seq techniques, which were developed in Richard Sandberg's lab and then further developed by Simone Picelli, among others. Our kit in particular effectively contains two modules to execute scRNA-seq. The first is a cDNA synthesis module, which contains all the reagents for cell lysis and reverse transcription, cDNA amplification, and cDNA cleanup. And the second is a library preparation module that contains all the reagents for TN5 mediated fragmentation and tagging, library amplification, and final cleanup. And we've kind of taken different approaches toward each of these modules when it comes to making the whole system more high throughput. For the cDNA synthesis module, we've leveraged the IDOT and CWASH from DISPENDIX. For the cDNA synthesis module, we've leveraged the IDOT and CWASH to automate the process and miniaturize reaction volumes in order to try and save on time and reagent costs and increase the throughput in setting up cDNA synthesis reactions per operator. For the library preparation module, what we've done is just implement this new ExpressPlex technology that Curtis just told you about, which also reduces hands-on and turnaround times for the library prep and has, I think, a lot of potential, still not fully realized, to enable much higher throughput at scale. So the first thing I'll be talking about is walking through results on our automating and miniaturizing the cDNA synthesis module. Before I get too much into the data, I just want to highlight a couple points on the experimental setup and interpretation. So first of all, in the experiments I'm going to walk through today, we've been using K562 total RNA as our control material. Each condition is tested in fairly high replicates. This is a type that's actually at least N of 6, mostly N of 8. For sequencing of the ExpressPlex and our standard library prep methods, all of that sequencing was generated in 48 plex multiplex samples or libraries and run on the NextSeq 2000, I think in this case. All of that data was targeted to generate at least 1 million reads and then sampled to 500,000 reads for analytical comparisons. Key metrics for performance for cDNA synthesis are really just yield and quality as assessed by bioanalyzer. And for sequencing, we obviously have looked sort of in the background at a number of different metrics for performance, but primarily I'll be talking about complexity and uniformity today. So the first step in working with the IDOT system and trying to automate our cDNA synthesis using this system is actually to train the instrument on accurate dispense of reagents that are required for these processes. So basically, the IDOT learns how much pressure is required to do its non-contact liquid dispensing and give the right volumes empirically. And this training can require anywhere from just a couple of data points to be generated to many, depending on sort of the nature of your reagents, things like the viscosity, surface tension, homogeneity, and so forth. Of course, we've kind of developed guidance for setting all this up with our reagents to facilitate any users who wanted to automate this kit. Once trained, we did find, as you see at the table at the bottom, that the IDOT shows exceptional accuracy and precision for both large and small volume dispenses, by which I mean, from microliters down to nanoliters, and when it's dispensing from large or small volumes. So when you have, a couple microliters in a well that is being dispensed all the way up to like 80 microliters in a well. After training the IDOT for use with our cDNA synthesis material, we use a device to execute a dispense of both our lysis and cDNA synthesis reagents for preparing these cDNA plates and cDNA synthesis reactions. And we compared the performance that we saw when we were doing this in an automated fashion using the IDOT with what we were able to get manually. We tested the process at a few different input levels that mimic sort of single cell-like masses of total RNA, as you can see here. And we found really no significant difference in the amount of cDNA that we were able to recover. And the bioanalyzer traces, if you're not used to looking at these, please believe me, they do look good and they look very consistent across both approaches. It's very good news. So it means we could save sort of time and hands-on energy by implementing automation IDOT. Using this automated cDNA synthesis setup system, we next evaluated the ability to use the IDOT and the CWash instrument to perform automated bead-based cleanups, again, to save more hands-on time primarily. So we leveraged the IDOT once again to dispense magnetic beads into amplified cDNA sample plates, which are sort of shown by this red plate here. After the IDOT dispenses those beads into sample plates, you can simply take your plate and put it onto the sort of balcony of the CWash for binding at room temperature. Once binding is complete, the CWash will just literally carry through an automatic wash and elution process. I will note here that during the elution incubation, we would typically remove the plate from the instrument and do a little bit of mixing off board. But then once your mixing and your elution is complete, the cDNA can be recovered manually with a multi-channel or using any kind of sort of common liquid handling equipment you might have, such as a nail drop or similar. So once again, this process was evaluated using both automated and manual preparations in parallel. And here you can see, once again, we were able to sort of meet or exceed the performance of our manual controls. So we got a good amount of cDNA generated in both of our automated cases, as much or more as we did manually. And once again, our bioanalyzer traces look very good, indicating that we had generated sort of clean cDNA libraries from this protocol. So having established these automated solutions for reagent dispense and cDNA cleanup, we next took advantage of the capabilities of these instruments, sort of the higher precision and lower volume working ranges, to miniaturize our reactions for the entire cDNA prep process. So we titrated our reaction volumes used for both lysis and then the actual cDNA synthesis reaction over a 10-fold range, ending at 2.5 microliter of total reaction volume. So that's sort of everything all in. Note that the primary limitation when you get down to that 2.5 microliter reaction volume actually becomes working with the lysis reagents. So the lysis has to be dispensed first, and then your RNA or cells need to go into that lysis. And what you can see on this table here is that lysis reagent comprises half a microliter at a 2.5 microliter total reaction volume. And of course, what that starts to mean is evaporation becomes more and more of a consideration. So we stopped at 2.5 microliters, although we got, excuse me, very good and consistent results at that 2.5 microliter level. So it's very plausible we could go down a little bit further. Interestingly, however, we found that as we sort of titrated to lower reaction volumes, we actually were not able to generate the same mass of cDNA that we observed at our standard 25 microliter reaction volume. This is shown in the plot in the upper right corner. You can see we got something like, say, about 70% of the cDNA back out relative to our sort of full volume reaction. And of course, being a bunch of problem solvers by nature, we immediately started screening a bunch of different assay conditions to try and essentially identify what the principal components might be that were limiting our cDNA mass yield. And then we performed combinatorial DOE to try and test and recapitulate the mass that we got in our 25 microliter reaction in these smaller volumes of 5 or 2.5 microliters, and those are shown in the red and the blue here. And if you look closely, you'll see we got pretty close, although I wouldn't say that we completely were able to recover that mass. But I'm primarily showing this to you mainly to convince you that we are aware of the phenomenon, and we have investigated it pretty extensively before I'm about to tell you that the phenomenon doesn't really seem to matter as far as information and complexity is concerned. So in parallel with doing these sort of principal component analyses to try and sort of add back to our reactions, we did, of course, take these 25, 5, and 2.5 microliter miniaturized samples, and we ran them on the sequencer. We prepped them with our standard library prep kit, and what we found is that we got essentially identical complexity, if not in some cases slightly better, at the lower input volumes. So 5 microliter and 2.5 microliter reactions, we saw literally superimposable gene body coverage uniformity. And again, I don't have time to sort of go into all the sequencing metrics today, but hopefully you'll believe me that this looked like a very convincing and comparable performance. Even though we weren't getting quite as high a mass of cDNA, it seemed like we were capturing all of the unique transcripts that we were able to capture, regardless of how much mass actually in the back end. So based on this series of experiments, we finalized an assay protocol for automated miniaturized cDNA preparation that is more or less described diagrammatically here. I'm not going to go over this ad nauseum, but I'd just like to point out that essentially you have 2 hands-on steps, a master mix preparation at the beginning, QC steps at the end, and that everything in between can be automated, and that this automation does in fact save you time, hands-on time, and volume, which also obviously translates to cost savings. So just to kind of give you some practical numbers around this, basically by reducing the cDNA synthesis assay volume to 2.5 microliters, you're able to fit enough reagent in an I.DOT dispense plate to fill 4 to 5 384 well plates, depending on your approach. This translates to about 1,900 cells that you're able to basically prepare cDNA from per I.DOT plate. It takes you about 1 hour to set up these 5 plates. It takes 2 hours for your RT-PCR reaction to generate your cDNA, and then it takes about 20 minutes per plate for your automated C-wash purification protocol. And so we think that you can very reasonably generate cDNA for 10 to 12 plates of cells per day, which translates to something like 3,900 to 4,600 cells per day. It's a very good throughput relative to what you might have been able to accomplish manually with a multi-channel. In the remaining 5 or so minutes that I have, I'm next going to just walk through some of the data we generated using the ExpressPlex module as the backend library prep for this automated miniaturized cDNA preparation protocol that I just showed you. So again, before I dig too far into the data, I'm just going to briefly turn back to the ExpressPlex library prep kit and remind us why this option is so valuable in terms of increasing the throughput of our RNA-seq solution. So as Curtis described, this ExpressPlex library prep module comprises what is truly an ultra-simple TN5 mediated library prep solution. And to use this solution downstream of cDNA synthesis, it is very simple. It requires only sort of 2 pipetting actions, again, moving material from an indexing plate into a ready reaction plate and moving cDNA material from a sample plate into that same ready reaction plate. And then once you have that ready reaction plate, you pop in a thermal cycler, you undergo a 60-minute thermal cycling program, you pool, purify, and sequence. And this whole process has about a 90-minute turnaround time for a single plate. And as you can imagine, because you're only really undergoing a couple sort of plate stamping actions, it's extremely easy to interface with automation, which is going to save you a lot of hands-on time. And I think importantly, even though this is a 90-minute turnaround time for a single plate, the low hands-on time and all that hands-on time being sort of at the upfront portion of the process means that staggered processing becomes very easy with this solution. So if you've generated your 12 cDNA plates the previous day, you can kind of very easily combine the materials for one ready reaction plate in ExpressPlex, one library prep, put it in the thermocycler, go back, do your next one, put it in the thermocycler, go back, do your next one, put it in the thermocycler. And we sort of conservatively estimate that I think you can easily process 8+ plates per day in this fashion. And then once all the ExpressPlex reaction plates are done thermocycling and generating their material, you can imagine that you can just pool all the individual plate libraries and then purify them all side by side. It's actually like a relatively simple workflow. To show that this technology can generate high quality cDNA libraries comparable to our sort of legacy module that I already showed you some data from, we first generated cDNA from K562 total RNA, once again using either our standard volume and manual approach, or we did it using this automated miniaturized protocol. So I'm referring to those as standard and mini in this slide, and we did this across several different RNA input levels. What you can see once again is that we lose a little bit of cDNA mass in our mini protocol, but that all the cDNA that we generated looks good, looks high quality. And so to run this as an end-to-end comparison, essentially what we did is we took the cDNA that we generated in our manual standard volume protocol, and we ran that through our standard library prep, or our legacy library prep. And then we took the cDNA that we generated using our miniaturized automated protocol, and we ran that in parallel through our ExpressPlex library prep kit. And I'm going to go on referring to this as standard and mini in these plots. And what we found sort of very gratifyingly was that when we did this end-to-end comparison, both workflows produced very robust libraries, and that we actually recovered much more material from a library perspective from our mini kit with the ExpressPlex than we did with our standard legacy kit, which is very cool. So we're generating a lot of material with that. Of course, the more important question here is really not how much library did we generate, but how much information is contained in that library. And indeed, we found that the complexities were, again, essentially identical across all different input levels, again, despite that difference in cDNA mass that I showed you, between both the standard and the miniaturized ExpressPlex reaction. This is a little bit hard to see, but similar to the plot that Curtis showed you earlier, we found that we got extremely consistent representation of all those different 48 plexed samples in this library. So everything sort of hovered right around the 0.9% representation or so, which was great, just sort of right in the target that we were looking for. And we also got very good gene body coverage uniformity. This is a little different relative to the plot that I just showed you, but you see, if anything, we actually find more coverage of the 5' end with our ExpressPlex solution. And then finally, we just took a little look into sort of the base composition of what we were able to recover with the ExpressPlex, and we found that, comparable to our standard kit, we see about 85% of the base calls coming from mRNA, as opposed to things like intergenic or intronic regions, and that basically we saw effectively no ribosomal RNA coming from the system, which of course is always gratifying. We don't want that sort of wasting our data. So having demonstrated that we could use this ExpressPlex workflow as a backend, we kind of once again sort of came up with this workflow method that's shown diagrammatically here. Not to bang away at this too hard, but I think it really is important to know how simple it is to combine these two plates, your sample and your indexing plate, into your ready reaction, throw it in the thermal cycler for 60 minutes, pool your library, which can again be accomplished using pretty common liquid handling technologies such as the L-drop, and then purify that library, and then you're sort of ready to go. So I think with that, just to kind of summarize, what we accomplished, or some of our conclusions, essentially using the IDOT and the CWASH, we were able to automate the workflow for preparation and purification of our cDNA. We were able to implement the ExpressPlex library prep module downstream for cDNA library prep and processing. Using this automated system, we were able to reduce the volume of our reactions by about 90%, which of course reduces costs and also allows you to sort of more rapidly dispense more plates. With the IDOT and CWASH, we were able to basically show or enable 10 to 12 plates of cDNA processing per operator day. The ExpressPlex, you can do 8+, probably library prep plates per operator day. And combined, this would reasonably allow you to expect a rolling analysis of at least probably greater than 3,000 cells per operator day, which I think is not the end of the road, but a great step forward in sort of translating full transcript RNA-seq into a little bit more of a high throughput